audio recording of the lecture 3A, virus structure and classification. Let's talk about the virus size. On the right-hand side of this slide, you will see a depiction of various molecules and organisms such as globular macromolecules that are in the range of 1 to 0.1 nanometer. Then we have viruses in the range of less than 10 nanometer, greater than 10 nanometer to about 1 micrometer. We have bacteria, eukaryotic cells, and lastly, the length of DNA genomes. So this is just a comparison of the size of the viruses with other structures. And on the left-hand side of this figure, you will see the range in which objects can be visualized by either light microscope, which is in the range of 1 millimeter to almost 100 nanometer, and below that, objects can be visualized by electron microscope. And as we can see, most viruses will fall into the category of being visualized by electron microscope. So overall, viruses are smaller than a cell, such as eukaryotic cells or bacteria, and they are larger than macromolecules. They're anywhere between 25 to 400 nanometer. They cannot be observed in light microscopy, and we can observe them. They are visible using electron microscopy. Next slide is just a depiction on the right-hand side of a gram-negative bacteria called Escherichia coli or E. coli, and a comparison of its size with some of the known viruses. These can include bacteriophages, F2, MS2, or even bacteriophage T2. These are viruses that infect bacteria or other viruses, and also comparison to vaccinia virus, which is one of the relatively larger viruses. So this slide just depicts a size comparison between bacteria and a series of viruses. Slide number 4 is a depiction of various RNA viruses. These are viruses that their genetic material is RNA. As we can see, there are different types of viruses, and these RNA viruses have different shapes. They can be helical viruses, or they can be icosahedral viruses. Slide number 5 is a similar slide to number 4, a depiction of various DNA viruses, those viruses that their genetic material is DNA. And as we can see, depending on the shape of the outer layer or the capsid of the virus, they can have different shapes. Slide number 6 is talks about the recently discovered mimiviruses. Mimiviruses are a genus of giant viruses. They're big viruses. In 1992, there was an outbreak of pneumonia in England, where the clinicians and scientists isolated a virus from an amoeba that was growing in waters of cooling towers. So virus was inside of the amoeba. Initially, they thought that this infectious agent that was isolated from amoeba. Initially, it was thought that it is a bacteria due its to relatively large size, about 500 nanometer, which is equal to 0.5 micrometer. However, they were unable to amplify bacterial 16S ribosomal RNA gene. This is a reference gene for degrees of homology within a genus. And since they were unable to find and amplify 16S ribosomal RNA gene, they concluded that the causative agent of pneumonia is not a prokaryotic cell. Future studies revealed that this Mimi virus is related to pox virus. It has a double-stranded DNA genome, and it's about 1.2 times to the power 6 base per long. Slide number 7 talks about the genome of eukaryotic cells. The genome of eukaryotic cells is double-stranded DNA. It's linear and is made up of chromosome segments, and it's roughly about 3 times 10 to the power 9 base pairs. Whereas the genome of prokaryotic cells is a double-stranded DNA again, but it's a closed circle. Unlike eukaryotic, that it has chromosomal segments, this is a single molecule and is significantly smaller, about 3 times 10 to the power 6 base pair long. Slide number 9 is a comparison of virus genome. Even though it's significantly smaller than both prokaryotic and eukaryotic genome, roughly about 3 times 10 to the power 3 to 3 times 10 to the power 5 base pair. However, it is a lot more complicated because it can be DNA or RNA. It can be single-stranded or double-stranded. It can be linear or circular. And lastly, it can be single molecule or segmented molecule, the genome. So we will describe all of these when we talk about various viruses. Slide number 10. This is a comparison of the genetic information contained in the chromosomes on the far right-hand side of the DNA of a human cell. It's comparable to numerous textbooks or encyclopedia, whereas the genetic information carried in the DNA of a bacteria, such as E. coli, would be comparable to the information contained in an encyclopedia. And lastly, the genetic information encoded in the DNA of a virus such as hepatitis B is very simple, very small amount, and it's comparable to one single sheet of information. Slide number 11 shows various classes of RNA viruses which have different types of genome, double-stranded, single-stranded, either segmented or as a single molecule. Slide number 12 is actually a depiction of the same concept where viral DNA genomes can be in different shapes. They can be double-stranded, single-stranded, segmented, or double-stranded circle or single-stranded circularized. Slide 13, viral capsid. Capsid is a protein shell that surrounds the virion, core, and nucleic acid genome of the virus. So it's basically a structure that protects the viral genome. One of a group of identical protein subunits that make up the capsid of a virus are called viral capsomer or protomer. So protomer or capsomer is one of a group of all identical protein subunits. And these are the structural units of the viral capsid. Viral capsid interacts with host cell. And a lot of times when the virus comes in, depending on their capsid proteins, such as the spike proteins of coronavirus, that will serve as an antigenic determinant, and the immune system recognizes those structures and will interact with them. So now that we're talking about proteins, let's have a little bit of a reminder of what we have learned in previous courses, and those are the 4 different structures of proteins. Proteins have primary, secondary, tertiary, and quaternary structures. The primary structure is just basically the unique order of these amino acids. The 20 essential amino acids are listed here. The amino acids that are linked together to form a polypeptide chain or a protein. And once these amino acids, or which constitute a polypeptide chain, coil or fold, they give the protein a 3-dimensional structure, and that is called the secondary structure of a protein. Whereas the tertiary structure of a protein is the entire and comprehensive 3-dimensional structure of the polypeptide chain of a protein. It can be either anti-parallel or beta-pleated sheets, which I'm sure you have learned in molecular genetics and chemistry courses before. And lastly, the quaternary structure of a protein is the structure of a protein macromolecule that is formed by various interactions between the multiple polypeptide chains that compose the protein itself. Slide number 17 is just basically a depiction of the same information that we just discussed. Slides number 18 and 19 are for those of you who may have forgotten about these various structures. So it's just here as a reminder of these protein structures. Slide number 20 talks about the shape of viruses. As we can see, viruses come in different shapes, and the shape is determined by the arrangement of the capsid proteins. Viruses can be helical, they can be icosahedral, or they can be complex viruses. And there are specific examples of each type of virus. Slide number 22 is the X-ray crystallography of icosahedral virus. As we can see, they crystallize the structure and they look at the details of the molecule, where each atom is located and where the bonds are. So let's talk a little bit about icosahedral viruses, their capsid and genome. As an example, we use poliovirus, and we discuss how the capsid of poliovirus is being assembled. Poliovirus has three structural proteins. These are called viral protein 0, VP1, and VP3. These three structural proteins mature and come together to form a structure called protomer. 5 of these protomers come together and form a pentamer. 12 pentamers, which is 12 times 5 is made up of 60 protomers, come together to form a procapsid, which is significantly larger than the pentamer and the protomer. And at this stage, RNA, or the nucleic acid, or the genome of the poliovirus, needs to be encapsulated, meaning that the viral RNA needs to become associated or with the capsimeres to form provirion. So procapsid, when RNA is encapsulated, will become provirion. And lastly, this VP0 structural protein will cleave to form VP2 and VP4, and these two proteins will interact with the virion. So a poliovirus fully assembled capsid has the RNA genome and has 4 subunits, identical subunits, VP1, VP2, VP3, and VP4. What about a helical capsid? A helical capsid is just basically a helical structure of the protein, or it looks like an elongated spiral cylinder of protein subunits or capsids where the genome is inside. And this helical structure or helix can be rigid or it can be very flexible. Slide number 26 is the depiction of a helical capsid of tobacco mosaic virus or TMV. The capsimer proteins, these identical subunits of proteins, are arranged in a circular disc. So each are just like a disc, and multiple discs will be stacked on top of each other, and they look like a cylinder shape. So we talked about how a poliovirus capsid is being assembled. Now let's talk about TMV as an example of a helical virus to see how its capsid is being assembled. Initially, individual and single yet identical capsimeres will associate with each other. Individuals will associate with each other to form a disc structure. And multiple discs will stack on top of each other to start forming a cylinder shape. And after multiple discs assemble and sit on top of each other, the disc forms a lock washer structure where the RNA will get stuck or will attach to that lock washer structure and will kind of, if you wish, will be sucked into this elongating helical protein structure or the capsids. So capsomeres come together, disc is being formed, discs will start sitting on top of each other to have multiple helical discs. Then the disc will translocate. It kind of moves a little bit around to form a lock washer structure where the RNA will be attached and RNA will be sucked in, and the helical protein structure will be elongated. Additional discs will stack pile on top of each other, and they will draw the RNA through the central hole. And this is the assembly of the TMV virus. Slide number 28, complex capsid. Some large viruses, such as pox virus, have additional layers and structures, which makes their capsid a complex structure. These additional structures and layers can be some structures such as surface tubule or core membranes. An example is depicted here in slide number 29, where we have on section A orthopox and on section B parapox. Both are part of the pox family of viruses. As you can see, they have envelopes, they have lateral bodies, and they have core membranes. These are additional structures that are being added to the virus and makes the capsid a complex structure. Let's talk a little bit about viral membrane envelope. As you know, the plasma membrane of eukaryotic cells is a lipid bilayer, two layers of lipids, where the hydrophobic groups are sticking outwards, and hydrophilic are sticking outwards, and hydrophobic groups are sticking inwards. And in between, we have integral membrane proteins. Some of them serve as ion channels, and some add a little bit of a structural stability to this bilayer. Pretty much similar concept in viruses, and the viral membrane envelope is a structure that surrounds the entire capsid of a virus, and this envelope is derived from host cell membrane. So let's see what will happen. When a virus wants to get out of a cell, it basically inserts itself and its glycoproteins into host cell membrane, and these glycoproteins interact with the host cell membrane. The membrane envelope that comes from host cell membrane can be dissolved by organic solvents. So once an organic solvent, such as ether, is being applied to the virus, the membrane will be dissolved. The virus is no longer infectious, cannot cause infections, and it is called ether sensitive. Slide number 32 starts by talking about the classification of viruses. Scientists by the name of David Baltimore initially classified viruses into 6 major classes based on their type of genome, and mechanism of messenger RNA synthesis. And this was based on the observations that the replication of viruses is dependent on their genome, and all viruses require messenger RNA in order to be able to translate their genome into viral proteins. So, Baltimore classification of viruses is based on the type of genome and mechanism of messenger RNA synthesis. And based on that, we have different classes, 6 different classes, which is depicted in slides number 32, 33, 34, or 33, 34, 35. These are all the same depiction. So let's go over one of these classifications. Class number 1 is double-stranded DNA viruses, such as adenovirus, herpes virus, pox viruses. Class number 2 are single-stranded, either positive or negative sense viruses, such as parvoviruses. Class number 3, double-stranded RNA viruses, reoviruses. Class number 4, single-stranded uh, RNA viruses that they can be divided into positive strand or negative strand. Positive strand can be viruses such as picorna viruses and toga viruses, whereas negative single-stranded RNA viruses or antisense RNA viruses such as orthomyxoviruses or rhabdoviruses. Then we have single-stranded RNA, RT viruses. These are positive strand or sense strand that their genome is RNA, but sometimes during their life cycle, we will see with the use of reverse transcriptase enzyme, they make DNA intermediates somewhere in their life cycle. Example of this would be retroviruses. And then there is also another more recently identified classes of viruses called double-stranded RT viruses. These are viruses that their genome is double-stranded, but they, as part of their life cycle, they use reverse transcriptase enzyme. Example would be hepatinoviruses, those viruses that cause hepatitis, and we will discuss them in depth and detail later on. Slide number 36 starts by talking a little bit more detail about various viruses in class 1, which are double-stranded DNA viruses. They have both sense and antisense strand of genome. Examples of viruses in this group are variola virus, the causative agent of a disease called smallpox, HSV or herpes simplex virus that causes cold sores. It causes lesions in both the genital tract as well as in the face and on the lips. It can also cause encephalitis, inflammation and infection of the brain, as well as STDs, sexually transmitted diseases. Therefore, herpes simplex virus, since we have lesions containing the virus in the genital area during sexual intercourse, they can be transmitted. So they're considered as sexually transmitted diseases. And adenovirus. Adenovirus causes respiratory infections and enteric or GI tract infections. This is the end of the audio recording on lecture 3A, virus structure.